Hello, Professor Gianneschi. It has been a pleasure to have you at the Department of Chemistry in Padua. We have a couple of questions for you.
Thank you. Yeah, pleasure to be here.
Nice. Can you please introduce us the concept of material biology?
Yeah, so there's really to some extent, a new idea or a way of formulating maybe a lot of work that's been happening over maybe the last decade. And asking the question really similar to what happened with chemical biology really famously over the last many years now. And that is asking, where they asked the question, can we develop small molecules to impact, to control, to perturb, to probe biological systems, in particular cells, in order to study biology? And so what we've been doing, and others too, has been asking this question of, what about at other length scales? What about, an example I gave, for example, today, as you know, is in the context of fibrillization or protein aggregation diseases, where they really are protein materials. So how do we probe, perturb, how do we disaggregate them, cause them to aggregate? So what types of games can we play that we've learnt from material science, in particular in our case, polymer science, to maybe develop new therapeutics and probes? And I think it's also, you applying those ideas to more like biophysics. So the concepts of polymer condensates or coactivation, fibrillization, self-assembly, supramolecular chemistry, non-covalent polymerization are these terms that kind of come from soft matter science and material science generally, to asking questions of, as I said, probing and perturbing disease-causing processes in cells, either to develop probes, maybe initially, as with chemical biology, but maybe also to develop drugs. And we think that there's a powerful concept there that has to do with matching the length scale. So if a protein interaction is occurring over long length scales, much larger than small molecules, how do we perturb and probe that interface? Well, maybe with drugs and probes that are the same length scale, so similar sizes, right? So that we have a chance of competing with those interactions. So that's kind of along the lines, if that's kind of clear of what we're thinking.
Thank you for your answer. And another question. What are the barriers that you face, the main barriers you face in clinical implementation?
Well, so this depends on the disease, of course.
Yeah, sure.
But in terms of the actual, selection of the therapy, this is true for any kind of medicinal chemistry program or therapeutic development. But I think more specifically to the types of things we've talked about here, and this idea of mimicking proteins in a new way, what you're doing is developing a new drug modality. And so you're not a small molecule, you're not an antibody, and so there's a regulatory pathway that might be new. There's certainly different types of questions that you're going to be asked. And that's really what, that discovery to development type program is what we're engaged in. But, I think that, there are that sort of new modality risk, if you like, right? If I'm going to do something completely new, then what are the other risks associated with it? It may not work, or there's some other toxicity you don't know about. But as I said, many of these interactions of interest, the protein targets of interest, are the undruggables or the difficult to deal with kind of protein-protein interactions that are very well known. It's that we didn't invent those. So I think it's that there's sort of the risk, but then there's the reward on the other side of that risk, which is that, can we find a way or a solution? And so we think that those kinds of risks of new modality are worth taking.
Okay. And talking about what you said before, talking about neurodegenerative issues and illness, what are the main illnesses that can be treated with this approach?
So we've tackled a few, and in the academic side, we have a lot of latitude to use different models. We've had different collaborators who have been interested in things like Huntington's disease. We've published on that. We've shown efficacy, for example, in a Huntington disease model in terms of the behavior of the animal, but also biochemical evidence that we're having a positive effect. But the disease model itself is complicated. It has some difficulty maybe in terms of translation. One of the really exciting, promising areas now that I talked a bit about was going after tauopathies, things like Alzheimer's, for example. And so we have a paper on the bioarchive now, and we're finalizing some more studies where we show that we can sort of play that game, right? We've got a polymer drug structure proteomimetic architecture that inhibits fibril growth. So can we then bind and then degrade the tau? We've shown that. So we've shown it in some robust ex vivo, so organoid type models of the brain. This is in collaboration with Ken Kosik's team at University of California, Santa Barbara. Nice. And so that's really the other fun part of that, right? These are people who are experts in the disease states, who are absolutely committed to going after that, let's say, that type of disease, and tauopathy in particular, and that disaggregation or the protein aggregation disease, and then we've been able to use this tool that we've developed to ask that question. If we mimic the sort of fibril forming pathogenic species, can we then break the symmetry of these fibrils and potentially inhibit or reverse the disease? So that's probably the most promising now. And I think if you look at it from the other perspective, there's obviously huge clinical unmet need. And again, a big barrier to other kinds of drug modalities. And so we're working on getting them into the brain, and there's some obviously big, really physical and biological hurdles. But the beginning of doing that has been really exciting.
Nice. We have the last question for you. Can you give us to the students, some suggestion to the student that would like to, try this approach?
Well, in my group, I can just say, well, when we meet new students, we've got them coming in the summer, and we have them come in the lab or an exchange, and they say, well, what are the projects you're working on? And I'll say, well, we've got these different things, and we like to make new molecules. We like to understand polymer chemistry, characterization, synthesis, designing probes, but they also get to work on cell studies and maybe take a material all the way through from that kind of design and discovery through to maybe developing it and applying it in robust models, working with lots of collaborators, as I said. And I think that's attractive. I think maybe that's always been the case. But certainly maybe more and more, there's this, they come in saying maybe they want to do something more translational. It doesn't always end up that way. And certainly there's a lot of hard work on the basic side. And I mentioned some of that, right? The physicochemical properties of the materials that some of them get engaged in. But I think it's attractive to students, right? To be able to go from synthesis, characterization, through to applying it maybe, and then asking and answering biological questions. I think that's really where they get excited. So I would say, whether it's me trying to get you to join my group, that's one thing, working on those problems, but also like I left it, I think there's lots of opportunities for material science principles to be applied in solving biological problems. And we do it one particular way, but you think of people developing new scaffolds, new implantable materials for healing, wound healing, for example, is a big area of interest. And so how can you develop more active sort of materials in the context of regeneration, for example? So I think students, maybe the ones I interact with at least, they're inherently interested in that kind of work. But I think also saying, hey, these are clearly unsolved problems. Yeah. Would you like to work on them, right? And learn how to make polymers while you're doing it. It's been an interesting and exciting approach. And also the students and postdocs did all of this work that we talk about. And invented a lot of it, right? They're co-inventors on these patents. And so there's also this opportunity, right, to engage in that inventive process, if you're at that cutting edge, and hopefully we are.
Thank you. Thank you so much for your time.
It's beautiful to be here in this amazing place and be able to talk to you guys, so thank you.
Yeah, thank you.